Hello, my name is Ani Michaud. I'm a 5th year graduate student in Bement Lab, and I'm here today to give you a little tour around the lab. So in the Bement Lab, we're interested in how cortical patterning of the cytoskeleton regulates two key processes, cell division and wound healing. We use Xenopus laevis, or the African clawed frog, to help us answer these questions. And we do this because they produce very large eggs, about 1 millimeter in diameter, and we can microinject RNA and protein directly into the cells and then image them on a microscope. So behind me, I have an example of some of the data that I've collected during my time in the lab so far. These are live frog cells that are expressing fluorescent probes targeted against proteins that we're interested in studying. For my project, I'm interested in studying these patterns and trying to determine how this patterning helps regulate cell division. All right, so now I'm going to give you a quick walkthrough of the rest of our lab. This is normally where Tom the postdoc sits, but since people have been a bit scarce lately, I had to make do with what I could. Let's turn it over to Tom and see what he has to say.
Hi everyone, I'm Tom and I hope you recognize me even without that righteous mustache. I'm currently studying the signaling mechanisms that both turn on and turn off the wound healing response in Xenopus oocytes. Most of my experiments consist of blasting frog eggs injected with fluorescent reporters with a special laser on a microscope to elicit the wound healing response. Not only do I get a lot of excellent visual data from the system, but it's as fun as it sounds.
In this room, we have Bill's office right over here. Hey, Bill. And then on this side of the room is where our resident scientist, Kevin, sits. Oh, it looks like Kevin's already in and working today. Let's see what Kevin has to say about his research.
My name is Kevin Sonneman. I'm a scientist in the lab, and I earned my keep around here by using the baculovirus system to express recombinant dysferlin and a couple of other proteins involved in membrane repair to both characterize their biochemical activities and also to visualize their involvement in the membrane repair process itself using the lab's frog oocyte wound repair model.
This is our cell room where we keep our frog cells and also where we microinject them with RNA and protein. And then over here, Zach is at one of our microinjectors getting ready to inject some embryos for his imaging experiment today. Here's our microinjector setup. We have a tiny glass needle that's sitting in this holder right here. You probably barely see it. And in this dish, there are some embryos, and the needle is hooked up to a microinjector, which, when you push a foot pedal that's underneath this bench, gives a tiny pulse of pressure, injecting RNA or protein into an individual cell. This is Bill. He's on our confocal system, doing some wounding of oocytes.
Basically, the idea is at the key time point, I'm going to switch the light paths, hit the laser, and then switch them back. There will be a wound. That's the wound. The yellow stuff in the middle is actually not very interesting. It's just burnt material. So what we're interested in is what happens around the outside of that burnt material.
Side note, our building is pretty old, so window access is rare. This desk right here is one of the only places in our lab that's next to a real window that gets actual sunlight. And it's currently vacant. You could sit here. Think about it. In summary, the Bement Lab is a fun and exciting place to work. All around the lab, there are little reminders to keep us motivated throughout the day and also promote good lab morale. We also do something called Spirit Week every year, which is a week-long lab event where every day is a different theme that we dress in costume for. Please enjoy some of these highlights, and if you're interested in working with us, don't hesitate to reach out. See you soon!